Every breath you take is an exchange with an atmosphere that is rapidly changing. And that changing air composition is forcing an invisible, population-wide shift inside the human body. For the last 150,000 years, Homo sapiens have evolved within an incredibly specific set of biological parameters. This chart shows atmospheric carbon dioxide baselines from Antarctic ice cores. For 800,000 years, ambient CO2 rarely exceeded 300 parts per million. On the far right, driven by industrialization, that concentration spiked exponentially, pushing past 420 parts per million in just 50 years. A 50-year atmospheric perturbation is a microsecond on the biological clock. Human evolutionary adaptation requires tens of thousands of years to account for environmental shifts of this magnitude. The standard assumption in physiology has been that our respiratory and renal systems can easily buffer this increase in ambient carbon dioxide. But this biological buffering is not free. We have to ask what hidden metabolic debt the human body is incurring to maintain homeostasis in this carbon-heavy environment. This atmospheric shift has become a metabolic one. It is now forcing a measurable reorganization of the molecules circulating in our blood. To measure this change, researchers looked at the U.S. National Health and Nutrition Examination Survey, or NHANES, the premier database for tracking population health. The scale of this study is massive. Using strict clinical methodologies, NHANES tracked the blood chemistry of about 7,000 people every 2 years between 1999 and 2020. The latest analysis of that data reveals a distinct shift in blood bicarbonate, in serum calcium, and in phosphorus. The biological community rightfully maintains strict skepticism here. A statistical correlation in population data does not automatically equal biological causation. To prove these changes are caused by the air we breathe, we must build a mechanical model of our cellular biochemistry. Every cell in the human body undergoes cellular respiration, producing energy along with a constant waste product that must be cleared, carbon dioxide. That CO2 diffuses out across the cell plasma membrane into the bloodstream. Almost all of it enters the red blood cells, known as erythrocytes. Inside these cells resides a critical enzyme called carbonic anhydrase. Carbonic anhydrase catalyzes a rapid hydration reaction, combining carbon dioxide and water to produce a hydrogen ion and bicarbonate. That resulting bicarbonate molecule is the body's primary transport mechanism for moving carbon dioxide safely through the venous system. Once the blood reaches the pulmonary capillaries in the lungs, the reaction reverses. The bicarbonate recombines with hydrogen to form water and CO2, ready for exhalation. Exhalation relies entirely on physics, requiring a steep diffusion gradient between the concentrated CO2 in the blood and the relatively clear air in the lungs. Today, the air filling our lungs is heavily enriched, exceeding 420 parts per million of CO2. Inhaling this elevated ambient CO2 flattens the diffusion gradient in the lungs. With less contrast between the blood and the air, it becomes physically harder to offload the waste gas. The immediate physiological consequence is hypercapnia, increased carbon dioxide retention directly in the blood. This retained CO2 drives up blood acidity, threatening to induce respiratory acidosis. To survive, the body immediately deploys systemic emergency compensation. The body's first defense against dropping blood pH falls to the kidneys. They increase bicarbonate reabsorption to buffer the acid and stabilize the blood. Under chronic, lifelong hypercapnia, renal compensation is eventually insufficient. The body requires a secondary chemical sink. That secondary buffer is the human skeleton, which can store excess CO2 deep in the bones. To temporarily stabilize blood pH, a chemical exchange occurs inside the bone matrix. Hydrogen ions are absorbed into the bone, and in return, essential minerals like calcium and phosphate are pushed out into the bloodstream. Chronic acidosis impairs the kidney's ability to process this freed calcium. Over time, it results in hypercalciuria, a net loss of calcium from the body through urine. Phosphorus is similarly drained from the body during this extended buffering process. The biochemical model is clear. Surviving a high CO2 environment requires a permanent increase in blood bicarbonate, paid for by the systemic depletion of bone minerals. We can now return to the NHANES dataset to see if this exact biochemical signature exists in the modern human population. This chart compares U.S. serum bicarbonate against atmospheric CO2. The top graph shows a clear upward trend in bicarbonate levels between 1999 and 2020. This 7% increase directly parallels the rise in atmospheric CO2 over the same period. This precisely maps to the modeled renal compensation for hypercapnia. Looking at the bottom graph, the solid line reveals the corresponding downward trend in serum calcium across the U.S. population. The dotted line shows a parallel 7% decrease in serum phosphorus over that exact same 20-year span. Because this dataset relies on massive sample sizes, the statistical variance is extremely low. This is not a clinical anomaly, it is a systemic event. The NHANES data provides the epidemiological proof. The skeletal buffering mechanism is actively occurring across the global population right now. The critical question is how long this physiological buffering can continue before the biological limits are breached. In human adults, the currently accepted upper healthy limit for venous bicarbonate is 30 milliequivalents per liter. If we project the NHANES trend line forward, the population average will cross into pathology, hitting that maximum limit, in the year 2076. Projecting the calcium and phosphorus depletion curves yields similar results. They will hit their respective minimum survival limits in 2099 and 2085. The first major clinical endpoint of this boundary breach is widespread calcification. The unmanageable mobilization of calcium leads directly to calcium carbonate deposits forming in the kidneys, arteries, and other tissues. This process is accelerated by the overexpression of carbonic anhydrase. The enzyme works in overdrive to process the excess CO2, which is heavily associated with dangerous tissue calcification. A secondary pathology develops inside our cells as endoplasmic reticulum stress. Elevated bicarbonate alters protein folding homeostasis, ultimately leading to organ malfunction. Chronic CO2 exposure also exacerbates oxidative stress by triggering the overproduction of reactive oxygen species, molecules that cause cellular DNA damage and chronic inflammation. The impacts extend to neurology. CO2-induced spikes in extracellular calcium restrict the movement of sodium ions, which directly reduces neuron excitability and impairs learning and cognitive performance. Within 50 years, the air we breathe will reach a chemical composition that human physiology is not evolved to process. Our current atmospheric trajectory is creating an unprecedented global public health crisis.